In this video, we will be talking about Chapter 11, the fundamentals of the nervous system and nervous tissue.
First, we will go over the three overlapping functions. Sensory input is when you gather information from your sensory receptors.
Then the nervous system processes and interprets sensory input and decides what should be done at each moment. This is called integration.
Following that, the nervous system activates effector organs, for example, muscles and glands, to cause a response called the motor output. Now we're going to break down the organization of the nervous system. The nervous system can be broken down into the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and the spinal cord. Its main function is to interpret sensory input and dictate motor output. Integration is the control center of the nervous system.
The peripheral nervous system consists of cranial nerves, which carry impulses to and from the brain, and spinal nerves, which carry impulses to and from the spinal cord. It also consists of ganglia, which are a collection of neuron cell bodies and act as communication lines between the central nervous system and the rest of the body.
The peripheral nervous system can be broken down into two functional subdivisions. The first subdivision is the sensory division, which is afferent. This consists of somatic afferents that carry sensory information from skin, muscles, and joints, and visceral afferents, which come from internal organs, as well as special sensory afferents, like our eyes, ears, and nose.
The second subdivision is the motor division, which is efferent. This means it transmits impulses from the central nervous system to effector organs, which are the muscles and glands. Somatic nervous system is composed of somatic motor nerve fibers that conduct impulses from the CNS to skeletal muscles. It is often referred to as the voluntary nervous system because it allows us to consciously control our skeletal muscles. The autonomic nervous system consists of visceral motor nerve fibers that regulate the activity of smooth muscle, cardiac muscles, and glands. The ANS has two functional subdivisions, the sympathetic division and parasympathetic. Sympathetic meaning fight or flight and parasympathetic meaning resting and digesting. Typically, these divisions work in opposition to each other. Whatever one stimulates, the other one inhibits. Now moving on to the histology of nervous tissue. A neuroglia is a supporting cell that allows neurons to do their job. The most abundant of the neuroglia are the astrocytes, which control the chemical environment. And then we have microglia, which are basically just fancy macrophages. Then we have ependymal cells, which form the cerebral spinal fluid in the CNS, and also oligodendrocytes, which form the myelin sheath in the neurons of the CNS. Moving on to the neuroglia in the PNS, we have satellite cells, which support neurons. Similar to the oligodendrocytes, we have Schwann cells or neurolemmacytes, which form the myelin sheath in the PNS. Moving on to some functional characteristics of neurons. They have an ability to conduct impulses. Neurons have extreme longevity, meaning they can function optimally for a lifetime. Neurons are also amitonic, meaning they don't have the ability to divide. Neurons have a high metabolic rate, which means they require a continuous amount of oxygen and glucose. There are three functional components of a neuron. First, they have an input region. Following that, they have a conducting component, and lastly, they have a secretory component. In the input region, they receive information through their dendrites and cell body. In the conducting component, they carry information throughout the axon. Lastly, in the secretory component, they release neurotransmitters through the axon terminal. Moving on to neuron anatomy, we first have the dendrites, which receives the information. This is known as the receptive region. The cell body of the neuron is also a part of the receptive region. The axon hillock of the neuron is where the action potential is generated. The Schwann cell forms the myelin sheath which wraps around the axon. The Schwann cell helps to make the action potential go faster. The axon then branches out into terminal branches. At the end of the terminal branches, we have the axon terminals, which secrete neurotransmitters. Now let's talk about neurophysiology, starting with the basic electrical principles.
Voltage is the difference in charge between 2 points. Current is the flow of electrical charge. And resistance is insulating ability to stop flow of electrons. Neurons have selective permeability of their plasma membrane, meaning they only allow certain things to come in and out. Now moving on to electrochemical gradients. First, there's the passive leakage channels, which is open 24/7 and allows things to flow through. Next, there's an active gated channels, which opens with a signal. The two types of the gated channels are the chemically gated, which is in dendrites and cell body, and they open with neurotransmitters, and the voltage gated, which is an axon and opens with a shock.
Now let's talk about resting membrane potential. A resting membrane is always at negative 70 millivolts and is ready to fire. It has unequal distribution of ions across plasma membrane, selective permeability of plasma membrane, and a sodium-potassium pump.
Now we'll be talking about the membrane potentials that act as signals. Depolarization is a decrease in membrane potential, which is the inside of a membrane that becomes less negative than the resting potential. Hyperpolarization is an increase in membrane potential. The inside of a membrane becomes more negative than the resting potential. Graded potential are short-lived, localized changes in membrane potential, usually in dendrites or the cell body. They can be either depolarizations or hyperpolarizations. In a neuron, an action potential is also called a nerve impulse and is typically generated only in axons. These voltage-gated channels are generally found only on the axons, where they are critical for action potential formation. No voltage-gated channels means no action potential.
During the generation of an action potential, we start with a resting state where sodium and potassium channels are closed and the membrane is set at negative 70 millivolts. During depolarization, the graded potential meets the threshold, which is negative 55 millivolts, and creates an action potential, making sodium pumps open and the membrane more positive or depolarized. In repolarization, the sodium pumps start to become inactive and potassium channels begin to open, causing the membrane to return to its resting state. In hyperpolarization, some potassium channels stay open, causing an undershoot, but eventually close, and sodium channels reset.
In this graph, it shows an action potential and how its voltage changes over time at a given point within the axon.
During the propagation of an action potential must be propagated along the axon's entire length. Action potentials can be propagated one of two ways. Continuous propagation is where the action potential in non-myelinated axon occurs. With south-story propagation, the electrical signal appears to jump from gap to gap along the axon.
Now we are going to talk about refractory periods. Absolute is when you can't generate an action potential because you're in the middle. Relative is when most sodium return to resting state and some potassium channels are still open as repolarization occurs. In the synapse, we have presynaptic and postsynaptic neuron. Electrical synapses are much less common than chemical synapses. They consist of gap junctions like those found between certain other body cells. Their channel proteins connect the cytoplasm of adjacent neurons and allow ions and small molecules to float directly from one neuron to the next. Chemical synapses are the most common type of synapse. They are specialized to allow the release and reception of chemical messengers, known as neurotransmitters. A typical chemical synapse is made up of two parts: a knob-like axon terminal of the presynaptic neuron, which contains many tiny membrane-bound sacs called synaptic vesicles, each containing thousands of neurotransmitter molecules. A neurotransmitter receptor region on the postsynaptic neuron's membrane, usually located on a dendrite or the cell body.
The organization of the synapse includes the axon terminal of the presynaptic neuron, which sends the information. The information diffuses into the synaptic cleft, which separates the two neurons and is around 1/1,000,000th of an inch wide. Receptors like the cell body and dendrites receive the neurotransmitters in the postsynaptic neuron. Information then transfers across a chemical synapse. Action potential first arrives at axon terminal. Voltage-gated channels open and neurotransmitters enter the axon terminal. Neurotransmitters diffuse across the synaptic cleft and bind to specific receptors on the postsynaptic membrane. Ion channels then open. Neurotransmitters then diffuse away from the synapse. Synaptic delays last 0.3 to 5 milliseconds, making transmission across the chemical synapse the slowest step of neural transmission.
At excitatory synapses, neurotransmitter binding depolarizes the postsynaptic membrane. In contrast to what happens on the axon membranes, chemically gated ion channels open to postsynaptic membranes. Binding of neurotransmitters at inhibitory synapses reduces a postsynaptic neuron's ability to generate an action potential. Now we're going to talk about the neurotransmitters and their receptors. The classifications by the chemical structure is acetylcholine, biogenic amines, amino acids, peptides, and novel messengers. Some neurotransmitters are excitatory and some are inhibitory. Others exert both effects depending on one specific receptor types before they interact. Neurotransmitters that act directly are those that bind to and open ion channels. Neurotransmitters that act indirectly promote broader, longer-lasting effects by acting through intercellular second messenger molecules, typically via G protein pathways. Now let's get into the neural integration, one of the basic concepts. Neuronal pools are a group of neurons that work together. The types of circuits are diverging, converging, reverberating, and parallel after discharge. I know we have only one job.